So maybe we can get started. So good afternoon, everyone. I'm Jose Moran Mirabal. I'm the Scientific Director of the Center for Advanced Light Microscopy. And the CALM has the privilege to partner with BIMR to offer this series of talks. And today, it is my great pleasure to welcome to this forum, Professor Pedro M Pereira. Pedro is a group leader at the Intracellular Microbial Infection Biology Lab, most micro at the ITQB Nova in Lisbon, Portugal. And so just a little bit of background about Pedro. He graduated with a degree in applied chemistry from Nova in 2006. And then following that, completed a master's in biotechnology. And within Nova, he also got his PhD in cell biology in 2013, where he started exploring not only microbiology, but also super resolution microscopy techniques. So that interest took him then to University College London, where he did his postdoc and focused on a number of different techniques that he's going to tell us about today, including super resolution, nanofluidics, software development, all very, very exciting. In 2019, he went back to Portugal, where since 2021, he has his own research group. And again, focusing on intracellular microbial infection biology at ITQB Nova. Is lots of really exciting work, and I'm really looking forward to this presentation. So Pedro, welcome to our seminar series.
Thank you very much, Jose. So first of all, thank you very much for the invite. I'm really happy to be here. So I'll start by sharing my screen and putting my talk up. So
Perfect.
as Jose mentioned, let me just actually put this like this here. Perfect. So again, thank you very much for the invite. It's lovely to be here. I would very much love to be there presential, but maybe at another time. So as I mentioned, I've been fascinated by microbiology and host pathogen interaction and the ways, the methodology you use to explore questions in that realm. So since my PhD, and actually since my degree, I've been exploring these sorts of things. And today I'm actually not going to talk to you so much about the biology of this, but more about the technological developments we have been, that I did during my postdoc. And I will give you some hints of what we are applying them to currently, but I'm more than happy to discuss the details afterwards. Okay. So a bit of introduction, just basically repeating what Jose mentioned. So I did my PhD at ITQB Nova, which is, the mayor of Oeiras will be very angry with me if I didn't say this, in Oeiras, which is in the greater Lisbon area, but I have to say Oeiras, although my university is in fact Lisbon. And then I went to, and there I was working mostly with cell biology in human pathogens. And that's where I first got acquainted with super resolution and did quite a lot of microscopy, FM, and so forth. And then this prompted my interest in this field. So I moved to UCL, to London, where I worked at UCL at the LMCB, at the Francis Crick Institute. Actually, not only applying techniques to host pathogen interaction question, but also developing some, because when you're working in the field, you tend to find out that the technology that's available really doesn't cut it sometimes to your specific needs. That's something I'm going to cover today. So the idea was always this. Currently now I have my group at ITQB, the Intracellular Microbial Infection Biology Lab, but I'm still focused on the same things, which is how pathogens interact with the mammalian hosts, and what cool ways can we use to study this interaction. So, and this is all around fluorescent microscopy, obviously, as you might have become suspicious by the abstract and my career. And the reason for this is very simple. Fluorescent microscopy was disrupted in cell biology. The capacity to image cells live and with molecular specificity, with molecular specific labeling, is actually completely revolutionary. So you can actually see what cells are doing live and actually probe specific parts of the cell and see dynamics and interactions and so forth. And what fluorescent microscopy always lacked was the capacity, the resolving power of electron microscopy, obviously, which basically covers this whole range of sizes in cell biology. And this is due to the diffraction limit. So there's this rule that we cannot see anything below roughly 200 to 300 nanometers. And this is where super resolution again brought another disruption, that we now have the advantages of fluorescent microscopy, be it molecular specific labeling and the capacity to do lifestyle imaging, but we can now reach resolutions close and in some cases actually as low as electron microscopy. Okay, and I'm going to tell you some details about some developments we've done in this area. Okay, but starting for the basics, what is the problem with this diffraction limit? So the problem is quite simple. If you have a GFP molecule, which you can see here, which is 2.5 nanometers in size, if you had no diffraction, the limit, you would see it has a perfect point source of light, and if you have enough photons, you would see it has a perfect point source of light of 2.5 nanometers. The problem is that you cannot see it with a naked eye. You actually need glass elements, you need a microscope, and this makes results in the fact that actually this beautiful 2.5 nanometer point source of light is now actually represented in your screen, because no one looks at the binoculars anymore, by this Eridix pattern that's roughly 250 nanometers, which if you're looking in a camera system, you see this pixelated image. And we know this very well. Actually, Anastabe showed this formula that you can actually know exactly what's the resolution you can achieve in your microscope, which is depending on our wavelength and your numerical aperture. And we know this quite well, but the problem is that sometimes we don't think about the limitations this brings. And the most obvious is, imagine that you have this structure here with these 8 molecules, and they're all separated by more than the diffraction limit. So you know that there are 8, and you see a square. Perfect. If only everything was like this, but it's not actually. So usually, and in most cases, what you see is that these 8 molecules are within the diffraction limit. And the result of this is that you lose all information. Instead of knowing that you have 8 molecules and a beautiful square, you have a dot which has all the information hidden within the diffraction limit. So, non-surprisingly, this resulted in the development of many super resolution techniques, which are basically techniques that try to overcome this limit by clever optical or analytical tricks. But all of them have one thing in common, or let's put it this way, one wish or one dream in common, which is to have an easy and cheap way of doing lifestyle imaging at the nanoscale. We all aim to do this. And obviously, we decided, or when I was doing my postdoc, we thought, okay, how can we help drive the community into this stream? So the first thing we had to choose is where to focus on. And for us, it was obvious that it would be single molecule localization microscopy, because it's one of the easiest techniques to apply, and it's the one that usually requires less expensive microscopes. So the idea was, okay, this is where we want to focus our attention, right? So, and today I'm going to talk to you about some developments we've done in this field. But first, let me explain you how this works. So how does single molecule work? So let's imagine we have this structure, whose internal structure is hidden within the diffraction limit. What we can do in single molecule is basically switch all the fluorophores off, and then stochastically switch on a small subset, which is spaced by more than the diffraction limit. And if you recall the square I just showed you, if they're spaced by more than the diffraction limit, you can very precisely detect and localize them in an image, right? So if you do this many, many, many times again, so you turn on a subset, you detect and localize them with a high precision, switch them off, turn on a different subset, and you do this again and again and again until until you reconstruct your structure with a 10-fold increase in resolution, right? So I guess the easiest way to do this, and now I will request the assistance of the public, is actually to play a little game to understand these concepts. So what I'm going to do now is ask you to basically switch on your microphones, and when I ask you, tell me your name, basically, all at the same time, okay? So when I say at the count of 3, you'll just all tell me your name. So 1, 2, 3.
Hey, sad enough.
So I have no clue what your names are. I'm sorry, it's very rude of me, but I didn't understand anything. But now if I do this differently and in a very weird way, Jose, could you please tell me your name?
Jose.
Perfect. Alex, could you please tell me your name?
Alex.
Perfect. So I know that Jose is called Jose, obviously, and Alex is called Alex. I know precisely what the name is. I can see where the sound is coming from. And what's the difference is that basically it takes me much more time to understand everyone's names, okay? So basically, this is the same concept as in single molecule. What you're doing is asking each molecule its name at a time. Okay, and basically what you're doing is you're trading, obviously that takes much more time. So effectively what you're doing is trading temporal resolution by spatial resolution, okay? And you have many clever tricks to do this. So you use photoswitchable fluorophores, you use high laser intensity UV illumination, some redox buffers, and then you have clever detection and localization algorithms to do this. Obviously, all of these things have disadvantages. Up front is the fact that it takes time. You have to acquire thousands of images to have your beautiful 10-fold increase in resolution, okay? And obviously, usually when you're talking about high laser intensity illumination in redox buffers, that's actually not very lifestyle friendly. So when I promised you that we're going to talk about lifestyle at the nanoscale, it's sort of not really fulfilling the promise, right? So how can we address this? The idea is simple. Maybe what we can do is target some of these issues that we have here, like the photoswitchable fluorophores that need UV illumination, or in storm that need high laser illumination, the toxic buffers, and so forth. So we thought, okay, so let's pick one, right? Let's reduce the laser illumination and see what happens. And what you get, actually, when we reduce the illumination you're using is that you have a massive change in the photophysics of your sample. If you're using really high illuminations, you have these beautiful single molecules blinking happily. But when you start decreasing your illumination, you start that you're having more and more overlapping fluorophores. And if you use these clever detection and localization algorithms, you see that you really require these single molecules to detect stuff, okay? Because once you start to have overlap, you start having more and more artifacts until basically you have a result that has nothing to do with your reality, which is a big problem if you're trying to understand biology, okay? So we thought, okay, so how can we actually address this? So the idea was, could we possibly decouple photoswitching from illumination intensity? So we need these things to blink, right? That's how we detect single molecules. But can we do it without basically blasting our samples with a lot of illumination? And our first idea was maybe what we can do is we find a system where we can use a fluorophore and some molecule like a quencher that will quench this fluorophore. And when it's closed, it's not emitting, but by some change in the environment, it would open and emit. So basically, this system would be like a spring-based system where only when the fluorophore is away from the quencher, we would have fluorescence, which means that effectively we would have blinking. Basically, what this means is that if this works, we have illumination-independent tunable photoswitching. So we thought quite a lot about this, on how we could do this. And for us, the obvious scaffold to do this was DNA. And this was because we remembered about molecular beacons. So molecular beacons are used to basically detect RNA molecules. And the whole concept is this. You have a DNA scaffold, which has a quencher and a fluorophore that's off. And only when it binds to the RNA of interest, it hybridizes, forces the quencher and the fluorophore to become apart, and you see fluorescence. And we thought maybe we can use this to our advantage. And obviously we can. That's why you see the name is Super Beacon, because it's super resolution, so we have to give it a really fashionable name. Has all other super resolution techniques. And we decided to find ways, a scaffold that we could use to make this happen. So what we did was design the loop that was non-hybridizable and a stem that would hybridize, and at the edge you would have a quencher and a fluorophore. And because it's DNA, we thought maybe by changing the temperature, the buffer, and the structure, we could have a mixed population, or different stages where all the molecules will be closed in the off state, a situation where they will be interchangeably between off and on states, and the situation will be completely on. So basically our idea was that we would have a way to control this photo-switching dynamics without using light, just by using temperature, buffer conditions, and the structure of our probe. So we thought, okay, this is a good idea, but how we go about in testing it? So the easiest way is to use fluidics, and I will go into that a bit further on, how another way of, and how fluidics are important for many things. But in this case, we just wanted to see how our system worked. So what we did was tether these super beacons to cover slip, and we went to the microscope. And what we see is that they blink happily. And you see there's a countdown there. What you'll see is that when that clock reached 0, we added formamide and all of our probes open. So basically showing you that actually we see this blinking behavior just because of the spontaneous opening and closing of our structures. What we can do with this is, highlighted here by these circles, is basically select single molecules and study their blinking statistics to understand their behavior and how we can better tune them to our needs. So what did we do? In this validation, we did many things. We measured what was the quencher role in photo switching. We tried to explore different structures and how they would influence photo switching. We changed buffers and temperature to see if we could make them blink more or less. But today I'm only going to focus on the quencher, because it's what really makes the fluorophore go dark, right? So for that, we used the same system as I showed you before, but this time we had 2 sets of probes. One which has the classical that I showed you, this DNA scaffold with a quencher and a fluorophore, and another one without the quencher. Basically has a control to see if the blinking we saw was actually due to this opening and closing of our structure. And indeed, when we did the statistics and the blinking statistics of these molecules, we saw something curious, which is 2 states. So if you look at this graph, which has laser power in the x-axis and off-state lifetime in the y-axis, you see that you seem to have, and you have our control beacons in blue and our super beacons in black, you see that you have 2 clear behaviors here. And actually, at first, this puzzled us, but then if you look at it, it's actually quite logical. What you have is 2 behaviors. One, which is in red here, that both control probe and the super beacon share, which is this short off state. And another one, which only the super beacons have, which is a long off state. And basically what you're seeing is the normal fluorophore dynamics, when basically fluorophores like, Alexa 647, for example, have the capacity to blink on their own. And this you see either when the super beacon is open, or if it doesn't have a quencher. But this is our super beacon behavior. So when you see, but this eventually bleaches, right? Like any fluorophore. But when it's quencher-based blinking, you have no bleaching. And while it's closed, you have a long off state, and it opens and you see light, basically. So this was quite excited, and I thought, okay, this works. And as I told you, we did many other things to validate this, but I would like to go straight to the end and show you how this behaves in the real world. Because at the time, we were very interested in understanding how viruses interact with cells. So I always have had the fascination by pathogens, be it bacteria, virus, or otherwise. And at this time, I was quite keen on exploring viral entry, viral infection. And one really cool thing is IFITM proteins. Interferon-inducible transmembrane proteins are a set of proteins that are actually broad-spectrum antiviral effectors, and they have the capacity to inhibit viral fusion by altering membrane fluidity. And one of them, IFITM1, is actually at the plasma membrane. So we thought, okay, maybe we can use our system to understand how this works. So what we did, which was a bit of a rough way of doing this, is basically prepare antibodies which are attached to streptavidin, and our biotinylated beacons are attached to streptavidin. So quite a bulky structure, but good enough to test our system. So what we wondered, and the real key question was, okay, so how is restriction happening? For example, we have, using as an example HIV, which is roughly 120 nanometers. Can the spatiotemporal organization of IFITM1 give us clues on how the restriction is happening? This is a classical super resolution question, right? How a structural organization can result in a biological phenotype. So what we did was very simple. We tried to explore how IFITMs were organized. And obviously, because we were interested in validating our probes, the first thing we did was to compare them with a classical approach. For this, we use classical immunostaining with a classical single molecule fluorophore, which is Alexa 647. But because our point was to prove that beacons can work at any illumination intensity, we did the imaging at low illumination, very lifestyle compatible, and very high illumination, classical storm illumination. So what we saw, unsurprisingly, is that if you image classical, if you do a classical storm acquisition at high illumination, you have beautiful super resolution results, you have beautiful distribution in the cell surface, and you see a cluster diameter of roughly 70 nanometers and a cluster distance of 120, which is very cool considering that HIV, for example, is 120 nanometers, but a bit more on that a bit later. So, but what we also saw is that actually, if you start decreasing laser intensity, images start getting very, very bad and very, very unlike what's reality, right? Or at least reality we can see with single molecule. So you see that actually cluster diameter starts to increase due to the loss in precision, and also cluster distances start to increase, which is due to the fact that you are actually losing a lot of information due to overlapping fluorophores. So what happens if we do this with beacons? Actually, it works beautifully. In reality, independent of laser illumination, we see the same behavior at all conditions. Basically, we have a cluster diameter of 80 nanometers and a cluster distance, mean cluster distance of 130, independent of the imaging conditions, which tells us one thing, that super beacons do work across illumination regimes. So their photo switching is independent of illumination intensity. And it tells us another thing, that actually our very crude system to test the beacons does really show in single molecule, because we do have bigger clusters and bigger cluster distance due to probably a bigger error of localization because of our much bigger system. But it doesn't matter, because what it allows us to conclude is that actually beacons work independent of illumination. But I've been telling you since the beginning that it's live cell, live cell, live cell, and I'm still not showing you live cell, am I? So obviously we went to do this in live cell imaging, because what we asked ourselves was, okay, this is all very good and dandy, but is this a static thing that's happening? It's like a static suit of armor in the cell surface, or is it mobile? Is this dynamic? And actually, if we use the same system with our super beacons and do lifestyle imaging, we actually see that these organization is quite dynamic, and you see that there are clusters that move quite fast and rearrange themselves in the cell surface. So actually what this allows us to conclude is that in fact this is not a static suit of armor. It's actually a very dynamic suit of armor that's in a cell surface, and it's basically patrolling the cell surface. And the mean distance between clusters plus the size of the clusters basically guarantees that no virus will go unseen into the cell. Obviously, viruses have other ways of overcoming this, but the cell really tries hard to defend itself against viral infection, okay? So what do we conclude from this first part? Basically, we aimed at developing a method, in this case, a probe, or in this case, a probe, that will allow us to do single molecularization microscopy at low illumination and tune the photo switching, okay? And in reality, what we did with these super beacons, and by this very simple design, with this loop that doesn't hybridize and this stem that hybridizes, is actually we obtain a probe that provides illumination-independent photo switching, offers tunable photo switching by structure and by buffer conditions, which we didn't show you, but I'm happy to discuss later on. It's completely independent from the fluorophore used, so it's all about the quencher versus fluorophore pair that you use, allows you to do live cell storm, and it's compatible with other DNA-based approaches. A similar approach was used, for example, in DNA paint, to have DNA paint with a probe that's basically fluorogenic, okay? So, and I've told you in the beginning that I was actually interested in bacteria, microbiology, and so forth. So where am I taking this? Where is my group taking this? So here the idea is very simple. So we are very interested in understanding the ultrastructure of bacteria and how this structure changes in different conditions. For example, antibiotics, in the presence of antibiotics. So what we're doing is actually using these DNA-based systems to actually create high-resolution structural models of bacteria and trying to explore how these change in different environmental conditions, okay? But I've been telling you about lifestyle imaging and how the dream is to have a cheap, easy to use super resolution method. But what I've been showing you is that you have to use some bulky antibodies, and you might imagine this is not the easiest thing to do, multicolor and so forth. So you still need to compromise. This is not a silver bullet, okay? As you might have understood. So what can we do? How can you do this without having to compromise? How can you have this protocol here, but then have your live imaging perfectly beautiful, and then your high resolution imaging in the end? Well, I'm sorry to say that the only way to do this is actually doing every single thing, okay? And the only way actually to do this is doing multimodal imaging, okay? So, and for this, our solution, rather than creating a really clever super resolution methodology, was to cheat a bit and allow you to do multiple imaging modalities in one experiment. For this, we developed open source and robust fluidics hardware and software system. And we call it Pumpy, which is our suite name for this system. But the official name and more serious is NanoJ Fluidics. And it basically has this design. What we did was design a series of Lego pumps, which are pretty robust, and they're controlled by Arduino. And we designed a software to control it via ImageJ. So what you effectively have is a very easy and cheap way of doing fairly precise fluidics. And I underline fairly because obviously this is not a professional syringe pump that will give you a precision of 1 microliter, but it's more than enough for most applications. For example, if you want to do fluid exchange to fix your samples, to add some compound, some antibiotic, whatever, it's good enough, I would say. And importantly, a professional in a very fancy syringe pump costs probably €1,000 or $1,000. And each one of these pumps costs like 30 or 40, something like this. And importantly, whereas a big syringe pump is like this big, these Lego syringe pumps are like this big. So you can have up to 128 working at the same time, which you see that if you're using actual really fancy syringe pumps, the prices would be kind of steep. And with this, you can actually parallelize a lot of things and automatize a lot of protocols in a very simple and efficient manner, okay? Just to show you how easy this is an image of one of our systems as mounted on a microscope. And basically what we're doing at this time, I'll show you, but we're basically doing live imaging and then fixing and then doing some fixed imaging. Okay, so as I was telling you, we can do quite a lot of protocols with this. We can do event-driven fixation, live-to-fix super resolution, multimodal super resolution. We can unsupervised live-to-fix, which was what I'm going to cover today with you. But actually, I'm happy to discuss all the other modalities and the other cool things we can do with this. And basically, when you have a robust fluidic system, the sky is the limit, and your imagination is what basically drives you. Okay, so what is this unsupervised live-to-fix cell imaging? The idea is get the microscope to work for you, right? And for us, the simplest test case was to get the microscope to identify a very simple transition, which is mitotic rounding. Obviously, this is very simple because the cell is spread and very much not round. And then suddenly, when it's going to divide, it rounds up. So analytically, this is actually quite easy to map, right? You have something that's really not circular at all. And then suddenly when they start to divide, it gets very circular. And when it reaches a threshold that you define, you said, okay, my cells are circular enough, fix and do something. Or you can actually change this and say, when my cells are circular enough, wait 1 hour for them to spread again, fix and do something. So in reality, you can do whatever you want. For us, it was very simple. We wanted to do a very simple assay that would allow us to do something that we would normally not be able to do. And for this, we did what? We have live imaging of some beautiful cells expressing tubulin GFP and H2BM cherry rounding up. And when they round, we fix them and then we stain them with something else. For example, here is actin, phalloidin 647, DAPI, and you can actually now see a lot of things that you wouldn't see before. So for example, here you see microtubular organization during rounding, and here you see the actin cortex all around the cell when it's dividing, right? So in reality, what you have is a very simple way of following a specific event through time. And actually, I can tell you for a fact that we basically pressed play and left home for the night, which is also quite handy when you're following events that take quite a lot of time, right? So again, this is very cool, but what are we aiming to use this for in the future? So I think it's very exciting to combine fluidics with this and unsupervised image with deep learning. And for me, which I'm focusing on host passage interaction and bacterial infection, I'm very keen to explore something we've just published, which is DeepBox. So it's basically using machine learning platforms to recognize bacteria and to understand what antibiotics are doing to bacteria, and also combine this with intercell division of bacteria. So the idea is to have a simple system which will allow us to test multiple conditions and get the microscope to identify specific events which we define and fix and stain and do whatever we want after these events happen, okay? So in conclusion of this second part, what we've done is basically developed a very simple system, and you can see here in this time lapse of we building the Lego pumps. They're very simple to build, not as fast as you see here. They take a bit more time than what you see here, but still, we have had many people building them, and they're very easy to build, and they're quite robust. Actually, now we actually evolved this a bit and have 3D printed versions, so they're a bit more sturdy, but still, they still go for Lego because the Lego has a high tolerance and is very precise. So these pumps that we use and these assemblies are actually quite robust to do these sorts of things. But basically, to finalize this part, what we aim to do was to create a system that would allow protocol automation in an easy manner and has everything as we did when I was in Ricardo's lab, which he still does, and I will always do it like this. Everything is open source, and everything is basically accessible to the community. And the idea is to actually allow the community to get multimodal super resolution imaging, live to fix, and this event-driven events happening. Yeah. So, but all of this, and mainly the super resolution part of this, does require you having a super resolution microscope, which is a bit of a challenge, right? Because they're kind of expensive. And they do require someone that knows what they're doing. And if you've ever done super resolution, you know that artifacts are dangerous and they can lead you down a rabbit hole. So this really requires you to have a fancy microscope to do stuff. And maybe I shouldn't have mentioned the artifacts because the technique I'm going to tell you about next also has some issues. Actually, it's quite robust, but people do tend to have a hard time believing some stuff. Not the technique itself, but some results. So you can do many things if you don't have a fancy microscope. You can do SURF, which is a purely analytical technique that allows you to do super resolution in any microscope. Basically, if you have a nice system with a good objective, you can use SURF, which is lovely. And Ricardo just published eSURF, which is the new iteration of SURF. So I'm not involved in that work, but I do recommend you have a look because it's very cool. Or you can use the technique I'm going to tell you about, which is expansion microscopy, okay? And this, because of the whole name, expansion microscopy, does tend to have people thinking, oof, that must be artifact land. But surprisingly enough, it's really not, okay? Or alternatively, before I move on, you can actually mix the two. And there was a recent bioarchives where X10, which is the tenfold, the expansion microscopy version that allows you, I'm still going to explain what expansion is, okay? So I'm just basically giving you some hints before I give you the demonstration of expansion microscopy. So basically, this one is a mix of SURF and X10 expansion microscopy. And what you're seeing here, believe it or not, is actually one antibody in a cover slip, and you can actually see the structure. So I do recommend you go check this paper and use SURF and do expansion. But again, I digress. What is expansion microscopy? And I think the best way to show you is with a demo. So you have this glove here, which has a cell drawn, okay? A very normal cell in a very normal size, right? So as you might have figured out, expansion microscopy, as the name suggests, has to do with expansion, right? So, and it's all that it is rather than finding some clever tricks optical tricks or analytical tricks to break the diffraction limit, you just put your sample above it. Just make it big, and everything you want to see is above the diffraction limit, so there's no more diffraction limit. It's lovely. So how does this work with our cell? Basically, if you expand it, oops, every time I try to do this, it fails and it's ridiculous, but I'll try it again. Aha! So if I really fill it up, our cell is now much bigger. So we can see a much higher level of detail in our cell just by making our glove bigger. And actually, this is all that expansion microscopy is about, is making stuff bigger. So it does entail a few steps, and it's not very live cell friendly, as in it's impossible to do live cells, so definitely not friendly, but it does allow you to do expansion. It does allow you to do super resolution up to 10 nanometers in resolution in a very simple microscope in 4 colors, and in any sample, okay? So what does this entail? Entails your normal sample, fixed, entails that you then stain it, and then you have to link it to a matrix. And this matrix, it's very simple. It's basically you polymerize in a very simple terms, you polymerize an STS page on top of your sample, you digest your sample so it doesn't offer resistance to expansion, and then you put it in water and it expands. And this magic, it's all based on a compound that's actually in diapers, which is sodium acrylate, which basically will function as a compound to absorb water and move and basically expand. And because your sample is embedded in this acrylamide gel, your sample will expand with it, okay? And the cool thing about this is that you can actually do this in any sample. So it has been done in yeast, has been done in parasites, in cells, this is a mitochondria, in classical stainings, in mouse brains, in Drosophila brains, in whatever you can think of, probably it has been done. I myself has done it in yeast, in bacteria, in cells, in Drosophila brains. So you can see that, and for example, Drosophila brains, I did it for kicks because a friend of mine wanted to see something. So it's a technique that it's actually quite easy to do. And the community led by the Boyden Lab is actually fantastic in sharing resources. So it's very easy to go immediately to the lab and start. This is how I started, and now I really like it and I use it for many things. So what can we do this? And what am I trying to explore in my lab using expansion microscopy? So as you might have figured out by the other applications that I want to do with the other things I showed you, I want to explore bacterial and host cell organization, okay? Preferably when they're interacting with one another. And for this, what I'm doing is actually doing expansion microscopy on bacteria. And what you can see here is a non-expanded versus the expanded Staphylococcus aureus. Here it's actually Shigella inside septum cages, and here it's just nuclear pores, okay? So it's a technique that you can use for many things, and we in the lab are aiming to actually use it to understand exactly what is the ultrastructural changes when we try to mess with bacteria and what happens when they interact with the host, okay? And I'm showing you this also in mainly this YouTube video because if you want to get started and you want to see me making a fool out of myself trying to explain it, I actually did the YouTube video going step by step on the protocol, trying to show you how I used to do things back at UCL when I was doing expansion microscopy, so I hope this helps. And also I've truly recommend you explore this expansionmicroscopy.org website, which is from the Boyden Lab, where they have all the protocols, all the papers, and it's fantastic. Actually, I was just in touch with the CEO of a company they're launching related to expansion microscopy because of this YouTube video. So probably it's not as bad as I thought. And on that note, I would very much like to thank you for your attention. I left plenty of time for question. I hope that's okay. And basically, I want to thank my team, which without whom I couldn't have done this. My collaboration, which were involved in this project one way or another. Funding, obviously. And if you by any chance know anyone wanting to come and do a PhD or a postdoc in Portugal, do send them my way. I'm very happy to host people, and obviously I would like to thank Ricardo Henriques, he was my supervisor during my postdoc. He's a lovely person, and if you want to basically explore machine learning, super resolution, he's the person to go to because he's a lovely human being. Thank you so much, and I'm open for all of your questions.
Thank you very much, Pedro, for that inspiring talk, and also very fun to watch, right? All the interesting things that you have developed. So at this moment, I'll open the floor for questions, and I see we have already some people raising their hand. So maybe we can ask them to unmute so that they can say their question or type it in the chat, and we're happy to read it for you as well. So Andre, please go ahead.
Yep. Hi, Pedro. Thank you so much for your presentation. It was really helpful. I actually also work with expansion microscopy. I had no idea that this was going to be a part of a presentation. So my first question is actually, have you ever tried a barrier star orange for expansion? Or
Okay.
do you know anything
No,
about
actually, I haven't. But
Okay.
I can reach out to the people, drop me an email, and I'll reach out because I know many people doing this in different systems. So
Okay.
I'm more than happy to follow up on that and see if someone has done it.
Okay, perfect. Yeah, because I asked Sven himself, and then he said that the star red is good, but not the orange. He has not tried it as well. And then my other question was that I wasn't exactly sure with the super beacon. Do you have like any idea what the linkage error of that may be? And could you couple that with like nanobodies by any chance?
Oh, well, at the time we did this because it was the most straightforward way of basically, we were focused on trying to prove that we actually could have illumination independent photo switching. But the beacons themselves, we have different sizes, okay? But yeah, DNA is big. So the linker error can be big, but not bigger than an antibody. So if what we would do is actually what your suggestion. So if we're worried about the linker error, we'll just fuse it to an antibody and use it like that. Because the big advantage here is that we can actually have it blinking without having to blast our cells. So even if the linker error was a huge problem, because we're dealing with live cells, we also don't have enough time to really reach the precision where that probably will start to be a problem. See what I mean? So yeah. Definitely it's a problem. I think for the applications we had in mind for them, it's not a deal breaker.
Mm-hmm. Okay. Yeah. Thank you so much. I will definitely send you an email. Thank you.
Perfect.
Thank you. Muhammad, you had your next question.
Thank you so much, Pedro, for the talk. This might be a bit of a silly question, but can we combine expansion microscopy with other super resolution techniques like STORM or SIM to get super resolution?
So the most, so there's loads of work on that, and people have been, so that's the beauty of expansion, is that you can actually very easily combine it with other super resolution techniques. Actually, one thing I did not mention, and it's probably one of the greatest things about the expansion, is that it's also a clearing method. So if you think about, for example, the caveats of going to tissue, but because light gets diffracted, you have aberrations. After you're done with your sample, it's water. So actually you can combine it with other super resolution microscopy techniques, and you have the advantage of your light going through and impeded. So you have an increase in resolution because of expansion, because you can use super resolution technique on top, and because it's a clearing method. So it's actually a beautiful technique. So for example, now I know that people have used STED with expansion microscopy, SIM with expansion microscopy, ARIScan, obviously, even STORM, which is probably the one that's the hardest because of what STORM entails. But with, for example, the X10, there reaches a point where if you try to go for STORM, you're already seeing the fluorophores. So it's the increase in resolution, it starts biologically, it just doesn't make sense anymore. So if you can do, for example, what we usually do is expansion plus SIM, so the twofold increase in resolution and the ease of doing 4 colors for us, or ARIScan, for example, is perfect.
That's amazing. Thanks so much. And it does look like, that's something that can unlock even sort of newer areas if you're reaching sort of single molecule sort of regimes, which seems to be the direction that SuperRes is sort of heading into.
And if I can add something to my reply, another thing that you can do actually, which is one of the big advantage of expansion also, is that you can actually stain after you expand. So imagine those really tight clusters of proteins and really tight structures, biological structures that your antibodies cannot reach. If you expand and stain after, now you can reach them. And usually this entails, it's not, so the homogenization part I mentioned, which is digesting your sample so it doesn't offer resistance to expansion. It can be done by proteases or by autoclaving or heat and SDS and so forth. And if usually when you stain after expansion, you use autoclaving or something that denatured proteins. And if you think about it, most antibodies are actually raised against peptides or unfolded proteins, right? So you not only get an increase because you actually make things further apart so antibodies can reach, but you're actually working with unfolded proteins at this time, or misfolded or not perfectly folded proteins. So actually your antibodies will recognize your proteins better. So it's actually win-win.
I guess that could also sort of help with sort of scenarios where you need a lot of localization density for sampling, and you reach a point where you have self-quenching when you have too many fluorophores in a small space. But then if now they're further apart, then that could help with that. I have another quick question. So you earlier at some point showed us this sort of lifetime of the fluorophores with under different illumination power conditions. Did you see that the lifetime, for the short off state changed with the illumination power, because you would expect this to sort of increase with higher powers? Is that something you saw?
So basically, in those specific conditions, we did this in many different conditions. So when you actually promote storm-like imaging regimes, or that you don't let the fluorophores bleach and so forth, you do see storm-like behavior. So you have fluorophores going on and off happily. So in the lower illumination regimes, you just have them basically still. I don't know if this answers your question, but we do see not a reduction in the off time, because you always have, so we did this in PBS, water, and basically storm buffers. And if you do this in water, you have a mix between fluorophores bleaching eventually. So that also skews a bit your statistics and your measurements, right? And when you're seeing at lower illuminations, you don't see basically a big change, maybe a slight change because of that. But I think the two factors are basically compensating for each other, if you know what I mean.
Yes, that makes sense. All right, well, thank you. I'll let others have a chance to ask questions. Thanks so much, Pedro.
Thank you.
Thanks, Mohammed. Cecile, do you want to go ahead and next question?
Thank you very much for a very interesting talk. I especially enjoyed the part about the molecular beacons. And there's something I was wondering is, how come this long off time you measure doesn't have an exponential statistic? It seemed to be like a...
I actually do not know why does it have. So the only reason, I see what you mean. The only reason I could say is that basically you have either in this specific condition, you have fluorophores that are quenched or fluorophores that are unquenched, right? So I think because of that, you might see that behavior in basically the open part of the fluorophores, right? So there's no intermediate behavior. You see them either always on or they're off, because I think now actually you caught me off guard because I do not remember what was the buffer we used on that one. But what you will see is that they are either closed or open. So what you see is very two strict populations, right? So you see the ones that are open and bleach fast, even though we are basically in some cases using low illumination regimes, and then they open, they take a long of time and they open, which in this specific case was probably due to the buffer we used, because we do see in different conditions the blinking happening. Another thing that we see, for example, is, and I think if we would tune specifically in that specific case, we were just seeing the behavior. So for us, for showing purposes, what So I think that probably what I should have shown you, which would be more in agreement with what you're asking, and rightly so, is where we played with the stem size. Because there you would have different blinking statistics, right? Here, what you see is, okay, we want to see, without a quencher, you have in that specific super beacon, without the quencher, you have one behavior, and then it's open always. And then with the super beacon, you have either open or closed. And probably that's why, but I do not remember what was the buffer. Probably we chose that one to show this because it's either in a condition where it's either closed or open. There's no intermediate behavior.
So this off time is more a photo bleaching time. It's like you lose them by photo bleaching.
No. It's actually the off time. So no, because that off time is measuring. So what we do measure is you identify something that's on, and then it's off, and it's on again. So what we're measuring there is 2 events. So, but what we're measuring basically is just those 2 events. So to have an exponential, we'd have to measure, basically, we'd have to have different events happening, and basically some would be on, for example, for, I don't know, 1 second, and others 10 seconds, and others, and then you would bleach. So you would see that exponential. Here we have these 2, basically they're on, they're basically then hybridized, open, they bleach, and that's it. Or we stop the imaging, which is also very likely in that specific case. Because in that specific experiment, what we wanted to show is this dual behavior, with quencher or without quencher. But actually, I actually do not remember how we did that specific experiment. I would have to go back and check. Actually,
no, it's nice. Very nice.
We have, so what I can tell you is that we do have, with, for example, with the different stem sizes, we do see that. We do see a decrease and a stepwise change.
Nice.
No, thank you.
Thanks, Cecile. I also wanted to make a small break because we're closing in on 3:30. That Tuesday we will have slots available to meet with Pedro. So if you want to continue this discussion, we will have some Zoom links available for you to meet with Pedro. So if you want to meet with him Tuesday noon onwards, our time, 5 p.m. for him. So very, very thankful for you to open those spots for us. Please contact Joao or myself, and we can schedule you in. So right now we have a question from Alex and Annika. Alex, if you don't mind, I would like to go with Annika first. Give the students a chance to ask their questions. Okay, so go ahead, Annika.
Perhaps we have the same question. So when you were mentioning the preparation of these samples, and you discussed with the expansion microscopy that there's protein unfolding that occurs so that you can look at these in more detail. Does this not disrupt the in situ protein conformation that you might be trying to study?
Oh yeah, very
Or
likely.
how do you get around that?
You don't. I don't think you do, actually. So, for example, if you're actually looking for specific conformations, I think but for that, for example, I would change the way you do things if that's the case. So, for example, you do not need to use an autoclave or 50 degrees with STS overnight, which are the classical techniques for that specific purpose. You can use, for example, classical immunostaining, and then you fix your antibodies, you cross-link your antibodies to your sample, then you digest, for example, and then your antibody recognize the protein on its original state, right? So in that way, you have no issue. Or you can do, for example, well, that would be the easiest solution, but you can always, for example, but that will not help you with conformations. Now that will be, you do your immunostaining, and if you're worried, for example, if you see, what I would do in that specific case would probably add an extra fixation step, in order, like you do a kin of a single molecule. When you finished, you do your primary, secondary, and then you do a secondary fixation to keep everything in place. And then that's what I'll probably do. So that way you're sure that your antibody stays stuck. And if you mainly, obviously, if your conformation, I'm thinking about a protein that's probably at a low concentration or something like that. Because if your protein that has a different conformation that you want to detect by immunostaining is super concentrated, you have no issues, right? Just do an immunostaining, it's fine. But if you do have, it's a very specific subset of proteins, I would do that. Use classical immunostaining, fix, and then do the expansion process.
Okay, thank you. That's really helpful. I had a follow-up question since it might be the last one here. So, have you seen any work done where there's just microscopy work exploring structural stability of viruses without cells? Like my project, for example, is looking at the immobilization of viruses in thin films. And so ideally, I would like it to look at the structure of the virus without having to transfect cells functionally. So, but it's a BSL-2 virus, so there's some difficulty in preparing samples for it.
So that's definitely. So I'm not in that paper, but it's so a great, marvelous friend of mine, fantastic person, very smart, much smarter than me, David Albrecht, which is now in Germany. He did the most complicated experiment that I've ever seen in my life, which was actually, I have in that paper now that I think of it, but which was to image viruses in a cover slip in 3 different microscopes, the same field of view of viruses. Okay, so imagine finding the same field of view in physically moving the sample to another microscope and doing this. And he did this because basically he wanted to correlate viral structure by different superresolution techniques. It's in the Squirrel paper from Sean Cully, another great person, by the way. Yes, definitely. So what we used to do when we wanted, so we did that for, I think he did that for vaccinia, but we did for HIV also. And the idea was always to fix and basically tether the virus to a surface. And then you can do any super resolution technique you want, expansion microscopy also. The only caveat you will face with expansion microscopy, which was what we saw with vaccinia, is that if your viral structure, I don't know which virus you're working with, but if your viral structure is very, very tight, like vaccinia is in the core, the idea of expansion is that you have to tether your proteins, and I didn't mention this, but you have to tether your proteins to the gel. And for that, you use a bifunctional linker, which has an NHS on one side and a acrylamide monomer on the other, okay? So if that molecule cannot penetrate your structure, when you pull, you're just pulling on the shell. So, and that's a problem if you have a very tight structure, okay? So, but if you just want to image virus with a superresolution technique, just tether them, and you can tether them in many ways. So if it's a virus you can change, you can input some, you can decorate with biotin and attach it to the surface. You can basically just let it dry on a surface. There's many, many ways. So David has that protocol in the scroll paper, how he did it. And vaccine is quite a big virus. So it's slightly different, it's like 500 nanometers. But yeah, I see no issue. And if you have any follow-up questions on that, because I don't know if I'm answering your question, drop me an email and I'll find stuff out for you.
Great, this is really helpful. Thank you.
Thank you, Annika. Alex?
Thanks, Pedro, for a great talk. I think you did already now answer my question, but I'll ask it anyway. And it was similar to Annika's first question. When you do this expansion microscopy, what I'm imagining just all the pieces of a cell or some structure just breaking apart, and then you just have like empty space between them because the gel is just breaking it into small fragments. And is that really what is happening? Or I guess you're linking it to the gel so it expands with the gel more smoothly, I guess.
It's only, I will just finish one thing I reminded Annika. You might also want to check Virus Mapper, which is something we developed when I was in the UK, which is it's single particle averaging without the hassles of single particle averaging. It's Fiji, open source also. And basically what you do is with your fluorescent microscopy image of your virus, align them all and create high resolution images of your virus with like single nanometer precision. If again, drop me an email if you have questions about that. More than happy to help. So Alex, yes, if it goes wrong. So basically the idea is I love expansion microscopy because first because everything is out there. So all the protocols it's all open source. That's the open source victory. But the thing is, when it goes wrong, it's very obvious that it went wrong. And exactly what you're describing. Basically, if you don't homogenize your sample properly, you see it break. And with cells, it's beautiful. That's when when I teach expansion, I always do it in the worst possible way, so that in the end I have those artifacts to show people. And it's beautiful because you see cells ruptured, like they were ripped apart. It's beautiful. But if you homogenize, so the theory behind it is you're using proteinase K, which is very promiscuous. So it will cut enough of your proteinase shift structure, thinking about the cell, obviously, so that you actually have isotropic expansion and you actually don't see all the breaks because you don't have enough resolution for that. When it comes to the X10, so this is because it's the 4.5 version. When it comes to the X10, so usually what you do to avoid it is, for example, imagine you are seeing a linear structure which is perfect for this sort of artifacts, like microtubules. What you would do, for example, is use alpha and beta tubulin raised in the same animal with a polyclonal secondary. So basically what you do is you're saturating your whole structure with, so you can even use primary antibodies labeled, secondaries labeled with the same color. So basically, or even if it's different antibodies, always the same color. So you decorate your sample so much that basically you again are within the resolution limit and you don't see it anymore.
Mm-hmm.
So you see a linear structure. Okay, but yeah, if you now go to the X10 and you are actually using just, for example, one antibody, I think you will see a patchy structure for sure, because then you're actually seeing how much you pulled stuff apart, right? But it's cool because you can actually use that in your favor. So has a control that your expansion went well. So if you know that your alpha tubulin has this spacing because the antibody is a monoclonal against that specific part of alpha tubulin, you can see, I see this spacing, I know that the antibodies should be X nanometers apart and I can measure it, which is quite powerful.
Yeah. Thank you. That's great.
Wonderful. Thank you. So Joao, I know that you had some question also pending there, so feel free to jump in.
Pedro, I'm just thanking you for a great talk. It was very cool. I'm thinking about your peristaltic pumps, your, how do you call it, pumpy? I mean, it's completely you can basically devise plans. Is it compatible with this autopilot from Loic Royer, from this auto source software?
That's a good question. I do not know.
And my point is, if that's the case, when can we start seeing self-experimental microscopes? Because I mean, this is completely, I mean, the idea is like we now have self-driving cars, right? So can we have like a self-experimental microscope in the future?
So you already have some people doing that, and not so the reason it's, I don't see why it wouldn't be. So the thing is that for example, every time we have had a request like that, it's because the code is open source. We've seen people adapt NanoGFLs for a bunch of things, and we have it working. So for example, in the system I showed you, it's with Micromanager, but you can use it with its own self-contained plugin or directly from Fiji. So I actually don't know, and I should, but I don't, to be honest. But I don't see why not, basically.
Exactly, right.
And the reality is, there's people like Chevarite is doing that with his single molecule 96 well plate automated microscope that basically does everything on its own. And as soon as you have machine learning working, if you have a very simple problem, you can do that. So if your question is cells divide, cells are basically rounding up, you can already do that for sure. If you have more complex problems, then you probably need a bit more work. But I know there are people already doing that, and it's
my question is more, are we ever going to stop needing scientists?
I hope not. I hope not.
Okay.
No, but I don't think so. I don't think so. Just for the simple fact that you actually need to go from tissue culture to the microscope carrying the.
Exactly.
Well, you actually don't, because you'll have a robot that does that. So the future is grim. That's what I have to say.
It's very grim. It's very grim. Thank you so much, Pedro. It was really cool. Thank you so much.
All right. With that note, I will end this seminar. Pedro, once again, thank you very much for a great seminar, very inspiring talk, lots of incredible ideas. I'll definitely follow up with you on some of them. And to remind the people who are still with us that we will have slots available to meet with Pedro next Tuesday in the afternoon. So with that, I'll thank everyone.